Here we are with nutrition and growth for the third topic that we need to know for the second exam. So the four main things we want to know here is more or less what is needed to grow, what happens when they grow together, how do we grow things in the lab, and then how do we measure that growth. So the idea behind this is reviewing what we have covered in the past, more or less the 5 properties of life, and one of them happens to be growth either in size or in number. So here we want to look at specifically how things grow in number more than anything else. So when we're looking to see how they grow, more or less what is needed and what happens when they do grow. So since we're going to be talking about bacteria more than anything else, when these guys happen to actually grow, they're growing in what we call colonies. That's assuming that they're growing from one single mother cell or parent cell, and they get to multiply relatively rapidly to grow into what we call colonies. In the event that we have multiple things going together at the same time, we usually end up calling those biofilms, and we'll explain those in a little bit down the line. However, what happens when we're looking at other types of organisms out there, like viruses? Viruses require a host, which then means that they can't quite grow in groups by themselves. So they get a slightly different name or title. So we call groups of viruses that have been developing inside a host or replicating. We call those a viral load, a viral titer, or a viral burden, depending on how you want to refer to them. So if you recall, when we went back to the first couple of topics, we were interested in what was needed to grow. And more than anything else, when we covered topic B specifically in chemistry, we looked at more or less the required elements of life. Now we're going to try and use them as a source of energy for things to grow. And then we're going to add, in this case, one primary compound for pretty much all life, which is going to be water, as well as whatever they need in terms of a location, a setting. This can be anything from atmosphere or anything in their environment. So let's go with the first topic here. What are the requirements or needs for something to actually grow and increase in number? So when we're looking at two key elements for growth, one of them is some sort of carbon source, meaning where they're going to obtain all the raw materials to build all their key macromolecules. And then the second point is where do they get their energy from? Typically this energy comes at least in some sort of origin as an electron. So we divide into two main groups our organisms, the so-called autotrophs, which use an inorganic source for obtaining their carbon, meaning something that it's never been alive at all. Typically this comes in as the shape of carbon dioxide, and then they utilize this carbon dioxide to form organic compounds. So they use something that is inorganic to make something that is organic. In other words, they're using the CO2 to build something like protein. Heterotrophs, on the other hand, what they're using is already organic compounds that have existed in the past, again, typically from living things, and they usually consume them, process them, and then they use those raw materials to build their own organic compounds. In other words, they already have to have organic compounds to build new organic compounds. That being said, once they have their carbon, in other words, their basic nutrient, they need the energy to be able to build new stuff. And so usually that means we have to find some sort of electron source. Autotrophs specifically, they also have to obtain their electrons from some sort of inorganic source. That means usually things like metals or inert materials. This sometimes can be called a lithotroph also. Heterotrophs, on the other hand, require their electrons to also come from those organic sources, meaning that whatever they consume has to provide them with that electron source as well. So the key feature here is that autotrophs get it from some sort of inorganic source, something that never has been alive, and heterotrophs get it from some sort of already alive thing or possibly dying. They consume it and then they can generate their organic compounds. Now, once they obtain those electrons, now they need to find a way to actually process that to turn it into energy. So, phototrophs is the type of organism that uses some sort of energy, usually radiation in the form of light, as a way to transport those electrons and move them around and then create energy, usually in the form of ATP. Chemotrophs, on the other hand, we're going to have to perform our classic oxidation reduction reactions more or less transferring electrons from one point to another, and that exchange of energy will eventually allow us the production of ATP as well. So here we have a slide that summarizes what we just covered in form of a table that compares both energy source and carbon source. For example, on the lower right hand side we have a group that represents animals, humans included, in which their idea is that they're obtaining both their carbon and their electrons from some sort of source that is alive or was alive. Conversely, in the upper left-hand corner, we have things like plants and algae considered to be photoautotrophs, and the idea here is that they're obtaining their carbon from typically carbon dioxide, something that is inorganic or never been alive, and their electrons are generated by the use of light or some sort of radiation, typically the sun. So let's take a look at the elements and some of the compounds that are required to maintain growth. Our first element that we get to observe is oxygen, simply because of it's one of the most electronegative elements that we have. In other words, it attracts electrons or it steals electrons quite easily. So in biology and microbiology as well, oxygen plays a major role as a terminal electron acceptor, meaning during metabolism, oxygen is used as a way to receive electrons while it's producing energy. Aerobes happen to be the ones that utilize it the most. They utilize oxygen as that terminal electron acceptor. That means that without it, they die. Anaerobes, on the other hand, use something else. However, in the presence of oxygen, because it can steal those electrons, since they're using a different one as a terminal electron acceptor, if oxygen is present, it actually will kill them. So oxygen is often lethal. So to create organisms that are more or less obligate, meaning that they have to have these strict requirements to have oxygen as a terminal electron acceptor or otherwise. And then we also have organisms that are called facultative. Facultative in this case meaning that some of these organisms have a choice, and then sometimes they can actually replace it. In other words, organisms can survive on oxygen, but when it's not present, they can choose to use something else as a terminal electron acceptor. Probably the best example here is E. coli, which thrives on oxygen, but in the absence of it, it can still survive, hence facultative. Other organisms that become aerotolerant, meaning that they cannot use oxygen, but in the event that it's present and it could harm them, they can actually remove it from their environment or detoxify it. Lactobacillus is an example for that. And then we have microaerophiles. These are organisms that have a very limited range on how to use oxygen. In other words, the oxygen that's present in our environment is around 20%. These guys require less than that, typically about 10% or less. In other words, anything above or below that range is usually harmful for them. Helicobacter pylori, present in our stomach, happens to play that role, for example, in which they require less than 10%. If you take it outside the stomach or in the environment that it has higher than 10%, usually Helicobacter pylori dies. So one of the easiest ways to observe the use of oxygen is by using a test called the thioglycolate test, or the thioglycolate medium, in which this solidified medium allows us to stab a tube that has virtually no oxygen present throughout the medium, and by observing where the growth occurs, we can determine what type of organism it is. For example, since at the top of the tube is the only place where oxygen is freely present, an obligate aerobe would be found on the top. But oppositely, an obligate anaerobe, which dies in the presence of oxygen, usually is growing way at the bottom of the tube. Facultative anaerobes, on the other hand, because they prefer oxygen, will survive at the top, but you can also see them distributed all throughout the tube simply because they can survive without it. On the other hand, aerotolerant organisms, since they prefer not to have oxygen, but in the presence of it they can detoxify it, can virtually grow anywhere in the tube. However, a common theme to test specific organisms like microaerophiles will use this medium to figure out exactly what their oxygen requirement is. Since at the top of the tube is about 20% oxygen in the air, and at the bottom 0, depending on the level of where these organisms grow, we can actually determine what their exact oxygen requirement is. Though often we see oxygen as this required element for life, more often than not, oxygen happens to be poisonous or even lethal to certain organisms simply because of its ability to steal electrons, that property called electronegativity. And so there are 4 levels of oxygen that we usually discuss that could be used either against organisms or simply they exist as a way to fight off organisms that can't tolerate oxygen. The most basic of it is singlet oxygen or good old oxygen gas, which for example our own macrophages, for example, will use inside their lysosomes as a way to destroy certain pathogens. Plants do in a similar way. They can actually remove this type of excess oxygen by using carotenoids, which are considered to be antioxidants. A higher level of a reactive oxygen species, or ROS, is a superoxide, in which this one having an extra electron kind of hanging next to it makes it more reactive and steals away other elements. However, certain organisms can protect themselves from it by using something called a superoxide dismutase, or a SOD, that will take that same element and now combine it and convert it into hydrogen peroxide. Hydrogen peroxide in and of itself can be toxic to certain organisms. However, this particular substance can be actually detoxified by certain organisms that possess catalysases or peroxidases, which are enzymes that are designed to break that down further into less reactive substances like water and oxygen gas. So this is something that we normally do in the lab by setting up certain organisms that we know possess these, giving them hydrogen peroxide, and then they produce oxygen gas, which in turn leads to this bubbling reaction that we can actually measure. So anytime people apply substances like hydrogen peroxide to wounds and we see them bubble, that's actually the organism deactivating it rather than having an effect. Now, probably the most reactive of the versions of oxygen that exist is the hydroxyl radical, the same one that we learn in acids and base reactions. That pretty much interacts with almost every single substance that we know and has a tendency of alkalinizing certain things, as well as producing bases that could be toxic to certain organisms. Another element that we usually refer to as essential is nitrogen. Nitrogen, happening to be this inert substance as nitrogen gas, is present at about 80% of our atmosphere. And since we need it for things like our proteins and our bases, it needs to be converted into a useful form. And so this conversion is usually referred to as nitrogen fixing, a process that we can't do, but there's plenty of organisms that can, specifically bacteria and certain fungi. And so we rely on these organisms to take the atmospheric nitrogen and convert it into useful forms. These are called nitrates and ammonia, more or less. And so we'll learn about which organisms actually do this during the classification lecture. So putting this all together, things like oxygen, nitrogen, we've already been talking about in chemistry and organic chemistry of hydrogen and carbon. These are more or less the key elements of life, including sulfur and phosphorus. But there's other elements out there considered to be trace elements that are needed in lower amounts as well. So the idea is every single bit of life needs these so-called 6 elements of life: carbon, nitrogen, oxygen, hydrogen, sulfur, and phosphorus. But there are certain metals out there and a couple of other elements that are needed in lower amounts. But the key ones are those 6. What about the physical requirements? So aside from the chemistry side to this story, we need to have more or less the environment play a role. The main factors that affect growth in this case, temperature being one. We've already discussed this plenty in the previous topic when we talk about the effects of proteins and denaturation, pH as well. But less often considered are the aspects behind water and its pressure inside the cell and outside the cell. So this is referred to as molarity. And then actual atmospheric pressure, so the pressure from the outside, things like the actual planet exerting forces upon you. Usually the very first physical trait that we observe is temperature. And as we've studied before during chemistry, we discussed that temperature is about motion. And the more things move, the more heat they generate. And so as temperature rises or lowers, it has an effect on cell structure. In particular, the membrane, for example, that is a lipid, and the more heat you apply to it, the more fluid it becomes. And similarly to proteins, that as their three-dimensional structure is dependent on more or less temperature, the higher the temperature goes, the more you can denature it. So usually we refer to the happy temperature that organisms live under as an optimal temperature for growth, but we also have delimiters above and below it that describe what is the minimum they can tolerate as well as the maximum. Typically, we can classify organisms based on some of the ranges of where they can survive. And so we have organisms that can survive from low ranges in temperature to high ones. At the lowest, then, we have organisms that are referred to as psychrophiles, which survive degrees that are usually between cold to freezing. So these are organisms that survive in things like the Arctic or on tops of mountains. Then we have mesophiles or mesophiles, which are us, which include within the range of temperature we are comfortable in. Above that, temperatures that are a little bit warmer or typically hotter are considered to be thermophiles. And anything beyond that range, we have what we call hyperthermophiles or extremophiles, organisms that survive at temperatures that are much, much higher than what we're used to handling. And so usually we can plot this as a spectrum and observe where this optimum temperature is, their maximum, their minimum, and we can grow organisms like bacteria to observe where their optimal growth is. Most organisms like us that are mesophiles survive roughly around 37 degrees Celsius. Here we can see a combined spectrum of the differences between psychrophiles, mesophiles, thermophiles, and hyperthermophiles. You can actually observe some of their overlap, but obviously their clear delineation of their optimal growth temperature. And so the idea here is to understand, depending on which organisms and their preferences are, we can learn how to treat them. Organisms like psychrophiles and thermophiles that do not live in our range are typically non-pathogenic to us simply because they can't survive under our conditions. However, other mesophiles, like ours, in this case, can harm us under our temperature, but if we take them out of that range, so we increase or lower their temperature, they can be controlled and typically even killed. Similar to this effect, we can talk about pH. Same idea as temperature, a physical environment, but here now we're talking about electrical charge, ultimately carried because of hydrogen protons. And the idea here is depending on how much or how many of those protons are available, depending on how acidic or basic, those will alter the electronic configuration of these molecules, and specifically, more than anything else, their charge. And so by electrocuting them, essentially, they alter the overall 3D structure of protein, breaking them down. So since proteins depend on their 3D structure, adding this acid environment or this alkaline environment will ultimately denature them. So we have also a scale based on organisms that survive roughly at neutral pH like us, considered to be neutrophiles. Then we have both acidophiles and alkalinophiles for their extremes. But then we can also have organisms that either are obligates, that require that situation, or organisms that can be tolerant to those extremes also. In other words, they can survive at a certain amount of pH for a certain amount of time and then return to their original state. Supporting what we covered in topic C when we talked about cell structure in the membrane and movement across from it, water plays an essential role for life. And more than anything else, having an isotonic environment is critical for more cell survivals, otherwise they either explode or shrivel and die. So the concept of osmotic pressure is also part of the physical environment that are needed. So certain changes in this environment can cause obviously effects on the ability to grow. However, there's organisms that can actually survive under much higher than usual amounts of solute present in their solvent. In other words, more stuff present in the water. And so these organisms are usually referred to as halophiles simply because they prefer higher than usual meaning here human, amounts of solvent. Similarly though, instead of the solvent, it could be simply the outside pressure that is present. So these organisms are usually referred to as barophiles. Assume that these organisms can now grow under these conditions. Certain combinations of them usually are disadvantageous because they're competing for the same resources. So this is called microbial antagonism, and something that we'll cover much later. However, every now and then these organisms can actually grow together and help each other out. And so most of the time we talk about symbiotic relationships, and we'll define that more carefully in the immune lecture. But every now and then organisms can survive together and survive better than if they were apart. And this is called a synergy. So when organisms can do this, we usually refer to them as biofilms, in which these organisms know how to grow together and actually can sense specifically how much to grow and when to grow, a concept known as quorum sensing, which allows them to maximize their resource use and even survive better. So now that we understand what is required for life to at least increase in number, we can actually master more or less how to do that in the laboratory. So the idea of culturing organisms. So this is pretty much the same material that we already covered in the laboratory, the idea of using a particular medium to help or increase the amount of growth of certain types of organisms while limiting others. So we have versions of what we call solid versus liquid media. We've already discussed those. We have media that specifically is designed to grow certain organisms versus others. We'll talk about that later. And so we had anything from Redox plates to simple agar to be able to grow the environment, whereas blood agar plates to grow things like our microbiome. But similar to that, the idea was certain organisms have specialized requirements. And so at this point in time, we try and learn additional materials that we can use to increase their growth, aside from the classic concepts of sterile techniques to limit the growth of any other contaminants present. So this includes streaking for isolation or purifying things within test tubes to actually serially dilute them and control how much growth we want. So now we will look at different types of media that are designed to support bacterial growth. Most of the time, the classic agar that we all hear about is a combination of sugars or polysaccharides that usually originate from algae. What's unique about this agar is the fact that it's liquid, usually above boiling temperatures, but solidifies below 40 degrees Celsius or so. So it becomes a very useful tool to be able to actually transport it and create different environments specifically to grow bacteria. Without this agar, you can actually include certain types of nutrients to actually grow the organisms by using just broth. And so what you add to that instead of the agar is using what we call peptone or beef extracts, so more or less liquefacions of certain types of proteins and other macromolecules that are sufficient to allow the growth of a particular organism. So putting chemical requirements aside, including water, and physical requirements aside too, we can create certain type of media that is designed to grow what we need. When we know exactly what is needed, we refer to that as a synthetic type of medium. Whenever the restrictions are not as strict in this case, we call it a complex type of medium in which we can mix things into it without knowing exact quantities. However, when we want something to grow specifically, we can actually limit the growth of something else or kill it by using what we call selective medium. One of the common ways to do that is by limiting glucose. Since it happens to play one of the major roles in providing energy, a lot of organisms will usually cease to exist without glucose. Often we don't really need to limit the growth of one organism rather than observe everything they can grow by actually differentiating them. And so we can create media that tells the difference between certain organisms and others while they're growing in the same exact environment. So we refer this as differential media. If you want to have certain organisms that require certain conditions, like an oxygen-free environment for our anaerobes or our obligates, we refer to that as reducing media. And for everything else, certain types of organisms have either time issues or very, very strict requirements. Often these organisms are referred to as fastidious. And so we create something called a transport media in which we actually use certain things like blood as well as certain environments to actually transport them or even grow them. So the example that we're seeing here is a type of differential medium, the classic blood agar plate that we've used in the past. But what we're seeing here is that different organisms, because of how they use the medium, change in color or what they actually display. So rather than killing one and leaving the other one to live, we can actually see their differences. Every now and then, though, we do have certain organisms that require a far more finessed type of medium to use. Some of the examples that include specialized media are things like viruses that require a host, or certain types of organisms like rickettsias, which are obligate intracellular pathogens, which means they require another cell to survive inside of. These require animal cells as host to culture. Sometimes some of those aerotolerants, rather than being completely devoid of oxygen, require a low oxygen condition, and so we create certain environments for them as well. Similarly, the opposite, sometimes they require higher amounts than usual, and these are referred to as enrichment media. And amongst our other examples, we have anything from organisms that are psychrophiles to thermophiles, and those utilize other environments as well. So what happens when we actually get them to grow, now we can actually measure them under our specific conditions. So similar to humans, by dividing by mitosis, most bacteria divide very similarly by undergoing binary fission. This is taking one cell dividing into two, two into four, and so on and so forth. So the pattern is pretty recognizable. However, one of the concepts that is important to see here is that by doing that type of growth more than anything else, is by doubling every cycle, so 2 to 4, 4 to 8, and so on and so forth, we encounter what we call logarithmic or exponential growth, which usually means that they can grow and divide and double extremely fast, as opposed to what we call arithmetic growth or arithmetic growth, this idea of 1 to 2 produce another 1, and so on and so forth, and this just adds and adds and adds in a very simple kind of straightforward addition. When we measure this, though, we can actually establish not only how long do they take to grow, but also how long do they take to die. So the entire cycle that we're looking at here includes the measurement of each division, and then we can divide it in a spectrum or a chart that describes the phases of them, in which stages in which they're not growing, called the lag phase. Usually that's preparation. Stages in which they are dividing, stages in which they've achieved a plateau, in other words, they're no longer starting to increase in number, and then some sort of period of decline or death. So to further explain this, we describe lag phase as the organisms obtaining the right materials and resources to begin to divide. Log phase or exponential phase, where the cells are actively dividing, that means replication and cell division. And then once they start running out of nutrients, that means that the amount of death and the amount of life being produced equal, so they enter this plateau. And then ultimately when those nutrients, macromolecules, run out, the organism itself begins to die. So this enters a decline or death phase. That being said, we need to accurately count how much of each organism is being produced during that specific log or exponential phase. There are two ways to do so, what we call direct and indirect methods, by looking at the actual organism growing, so by counting it, so this is a direct method, or by observing some sort of trait about it, something that gives us an estimate of what could be growing and how much of it. So these are indirect methods. So we'll look at them all briefly to kind of dictate their differences. So we can look at the first set of methods by observing the ones we count actively as they grow. The most common example of this is literally looking at a plate and counting. Sometimes when there are too many to count, the idea is to dilute them first and then count and estimate from there. So here we observe every individual colony as representing the original parent cell, and so by counting each individual in there, that represents how many original cells were in that original sample. So by back calculating it, more or less doing some simple math, we can estimate the original sample and how much was present in there. When the organisms are too few to count, sometimes we can concentrate it by using a filter. So by passing it through a small filter that doesn't allow the cells to pass through and concentrating the media, then we can replate those and then again remember to adjust for the calculation to estimate how much was there originally. At times, plating is not necessarily the best option, especially from liquid environments. So examples for water testing and water quality usually use this method in which you take a sample, serially diluted 1 to 10, and the idea here is to actually take multiple samples of them, and by seeing how many of those actual samples grow, by counting which of the tubes have growth and which of them don't, we can utilize a table that by using a little bit of a statistical analysis allows us to back calculate which is the most likely concentration present within those samples. Now, there are certain direct methods that still count actively, however, they do not require the organism to be growing or in some sort of environment. The most obvious example of direct counting without incubation is using a cell sorter or counting chamber, in which we take specialized slides, usually made out of quartz, that have within them an area that has been already delineated to encompass a certain volume. And so because we know the area and the volume directly associated with it, as we count certain cells, we can estimate how many cells are present in the original volume. A more advanced version of this is by using an electronic cell sorter or an electronic counter that usually uses light as a way to count individual cells as they are passing through a chamber. These are often called flow cytometers, and even then we can actually differentiate multiple types of cells by using fluorescence. Each cell is tagged differently, and so by passing through individual beams of light, or typically lasers, we can make those cells fluoresce, and each cell can be counted separately, pretty quickly, accurately, usually in the trillions within about 5 minutes. Moving on to indirect methods, in which now we're using an estimate of what is growing there by not counting directly, obviously. The most common way here is to establish how much are they consuming. In other words, how much are they eating and how much energy are they actually producing. So this is referred to as a metabolic activity assay. This is something we're going to do in the lab. Different way to be is to take the volume of these materials and then desiccate them, dry them out more or less under heat. And then based on how much dry weight they have, we can calculate how much is there. Probably the most common example that exists is the use of a spectrophotometer by using the original chemistry calculation called Beer's law that establishes how much is present based on how much light can pass through a sample that originally was transparent, meaning the complete amount of light that could pass through versus how much light can no longer pass based on how much cells are growing within that material, in other words, turbidity. So the spectrophotometer measures how much is transmitted and how much light is absorbed, and that usually tells us a definite estimate of how much is present. And then the last method, a more modern method of indirect counting, is by using the calculations of DNA by amplifying this, a protocol called PCR that tells us how much DNA was originally there, and that allows us to know how many cells were there present early.